Good evening. This is Thursday, October 15th, and I'm going to introduce you to the protein modeling kit or the amino acid starter kit. It was its official name. This is all about the levels of protein folding. And so this is hopefully going to help you work through the modeling activity handout that is in Google Classroom. Unfortunately, it would be great if you could actually do the model, but in this case, we'll just have to settle for doing it visually. So there's some basic pieces. There is this tuber, we call it a tuber. It's just a piece of foam with some wire in it so it can be bent. And this represents the backbone. This represents the backbone of the protein, which the backbone is made up of the primary structure. It's the stringing together of the amino acids. You may recall, I'm just going to show you this picture. This is a typical amino acid. And you can see that it's got the alpha carbon, or which is also called the central carbon. There's an amine group to the left. The red group to the right is the carboxyl group. So that makes it an amino acid because of the carboxylic acid group, amino acid. And then where the green star is, those are the side chains or the R groups. And down below, you're seeing two amino acids strung together. And what happens is you pull out water, dehydration synthesis, and you form a bond between the carbon of the one amino acid and the nitrogen of the other amino acid. That's the peptide bond, and that forms the backbone. The backbone is this repeating chain. Sorry, I'm trying to do this backwards. Is this repeating chain, if you look at it, amino group, central carbon, carboxyl group, amino group, central carbon, carboxyl group, amino group, central carbon, carboxyl group, amino, central carbon, carboxyl group. That's what this is representing, is that primary structure. And we have the blue end represents the amine group, and the red end represents the carboxyl group. So it makes a difference how you orientate your tuber. Right? It makes a difference. And unfortunately, it's showing up different on the screen than it is in my hand. But you'll get the idea. So this is the tuber. You'll also have this chart. I've attached pictures in the assignment folder or assignment in Google Classroom. And this chart shows you all 20 amino acids, and it shows you the color coding of the clips and the balls in the kit. Again, those are all available in photos. And then this is the placemat, which has already been put together by another group of students. And this contains models of all 20 amino acids. And the colors are not accidental. Each color clip represents a different property. So the white clips are what we call hydrophilic, water-loving. The ones with red clips create acidic properties. The ones with blue clips create basic products. And the ones with the yellow clips represent the hydrophobic or nonpolar amino acids. And there's two right in the center you might notice have green clips. Those are called cysteines, and they contain sulfur, which you're going to see plays a key role in protein stability. So students first had to take the clips and the amino acids and using the chart, match up the amino acids to the right clips and place them on the placemat, so that we had a view of all 20 amino acids. Again, I've got photos of each section in the assignment as well. So let me just share my screen and point that out to you a little bit more clearly. Whoops, I don't want to show you this young man's personal information. So here we are in Google Classroom. I got too many screens open. Go to classwork. Hopefully you already saw. I need to go to AP Bio. Hopefully you already saw some of the video help. These are, what I'm realizing is you guys are missing a lot of the conversations that happen between activities, before activities, after activities, that really provide the information to help you connect the activities to the content and help you connect the concepts across the whole idea. I'm afraid you might be missing how all this all fits together. So I'm hoping those videos will help. I strongly encourage you to listen to them. This is the video and photo help for the protein modeling kit. And as I said, you're going to find pictures in here. So there's pictures of the charts so you can look at them and enlarge them. There's pictures of the placemat. So here's the whole placemat itself. Of course, you can zoom in, look at things more closely. Again, each colored ball represents a different key element, et cetera, et cetera. If you use the directions that are in the irregular assignment, this goes with, by the way, this all goes with the Amino Acid Kit 1. I will be making a separate video for Amino Acid Kit 2 as well, but I'll put it in the same folder. Right, so just be realized those are there. So what students are instructed to do is to take this tuber, which is the primary structure, and pick so many of each amino acids. It's up to the students to pick which amino acids they're going to put on the tuber. The clip represents the bond between the central carbon and the R group, and then this is the actual R group. The R groups are the only part of the amino acid that are different. Every amino acid has an amine group, a carboxyl group, and a central carbon, and a hydrogen. There's only one bonding site where it differs, and that's the side chain or the R group. So here I've got two different side chains. This is a hydrophobic side chain, which is going to make this amino acid act as a water-fearing component. This is a base. This is going to make this act like a negative region on the amino acid. And so students, using some criteria, as you'll see in the worksheet, picked a series of amino acids and laid them out on the tuber. All right, I'm going to step away just for a second and grab one that students completed the other day. Okay, we're back. So here is one that a group of students assembled on the last class period that AP was together, which was last Monday. The blue cap must still be on their table. You can see the red cap. This is the carboxyl end. This end should have the blue cap, which is the amine end. The amino end always goes to the right, which on the screen, I'm not sure that's going to work. All right. So then what they had to start doing was figuring out, how would you fold this? How would you fold this? Well, hydrophobic molecules don't like water, so where would they want to go? They're going to want to bend inward and away from the water. So I start taking all my yellow ones and start pushing them to the inside, but the white ones are the opposite. They want to go outward. So they're going to want to push outward. The yellow one's going to want to push inward. I'm not sure you can even see what I'm doing, but you can start to see I'm trying to get all the yellow ones to bend inward because they're hydrophobic. They're going to want to be away from the water. The white ones are going to go to the outside because they like water. Now here, this acid is positive and this base is negative. So positives are going to attract to negatives. So that's going to put a kink right here. The two green cysteines with the sulfur are attracting. That builds something called a disulfide bridge. This negative is also going to be attracted to a positive. And what I'm trying to do is fold this to meet all of the rules as best I can. So all of these yellow ones are going to be stuck to the inside. The positives are attracted to the negatives. The disulfide bridges are together. And what you find out is you can meet most of the rules, but there are some compromises. There are some compromises. And this molecule, sorry, I dropped the piece. This molecule is now taking on a three-dimensional shape. And it's not just a random shape. What's dictating the way it's folding is how the R groups are interacting. This attraction between the positive and negative is pulling those together and putting a kink in the molecule. This disulfide bridge, which is a very strong bond, is going to pull those two pieces together and keep that very rigid and tight. These yellow ones are going to try to tuck inside here and stay away from the water. That's determining the three-dimensional shape. And remember, of course, the shape determines the function. So for you guys, this is the one you'll use to draw on your paper. I'm just going to spin it different ways. You can freeze the video at any point and use this to make a sketch. You're also, I'm going to pull it back into its before it folded, because you're going to want to list the sequence. And so I'm going to carefully move these. I'm going to start with the amine end and move this along. Okay. This will help you identify which is which. So this has got a white ball in it. Then there's the gray ball. Then this is the next one. Then there's this one. And then there's this one. And there's this one. Next. Next. And next. I'll try to spin them so you can see them. Next. 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 This is the primary structure. This is what's built at the ribosome. We skipped over secondary structure. We're going to do that in part 2, and we went straight to tertiary. So when I was folding it, that was modeling the tertiary structure, the third level structure. That's the most critical because that's when you create the three-dimensional shape, and it's the three-dimensional shape that determines the function. So the key is what we want you to do is to see how the placement of the R groups, the sequence of the R groups, totally determines the folding pattern, which totally determines the function. If you start changing these R groups, you're going to change the chemistry, and it's going to start to cause it to fold differently. And if you change the wrong groups and it folds differently in the wrong place, it no longer does its job or does something completely different. So I'm hoping this gives you enough that you can work through the handout for Amino Acid Kit number 1. I will try to post number 2 fairly soon. Have a good night.